Yeah. So title already everyone know, and as you people can see that our mucosa cell surface is so broad. So I will discuss those things as well. But what happens in mucosa? There is a microbes, especially in the gut. Gut microbes is present. Immune system like T helper cell is present, and in microbes there is a two type of microbes, like kind of commensal or kind of the pathobionts. Their interaction make T-Rex cells, and from microbes there are several components secreted outside, like CSGG. This is a kind of a glucoglycan. So interact with our immune system, mature the immune system, generate the T-Rex cells. These scenario make the tolerance, and during the tolerance, our immune system develops some TCR repertoire as well. You can see, but today I'm not going to show the TCR repertoire data. So there are several reports on that. So, so like you can see, when the baby born come in this world first time, so they encountered by several microbes. They can be bacteria, viruses, fungi, and sometimes like protozoans as well. So this is the first time introduction of the microbes on the kids, or any this rule apply for the any animal as well. So they get plenty of niche to colonize on the host. So what happened during colonization? There are two type of bacteria can colonize: good bacteria and bad bacteria. So these good bacteria called usually commensals, bad bacteria called usually pathobionts, or they are living together but they are not making harm sometimes as well. And good bacteria is responsible to mature our immune system, kind of anti-inflammatory responses. Bad bacteria is giving the pro-inflammatory responses. So what are the parts in our body for the colonization? So like you can see, nose, where we inhale anything, mouth, we start to eat, lung. So we inhale several type of microbes from the environment. So lung is also having the colonization of these microbes. Then comes gut bacteria. So gut bacteria is usually coming from the food, whatever we are eating. So that's why people say that good food is good health, and bad food can have the bad health. Sexual organs, skins, and this. Ah, my slideshow is gone.
हाँ हाँ हाँ, उसको नहीं, स्लाइड दिखाएंगे तो आ जाएगा। वो है, स्क्रीन है ही, जस्ट शो योर स्लाइड। यस,
हाँ हाँ,
इसमें
आया
आ
ना।
गया, आ गया,
एक्चुअली
आ
उसमें एक
गया।
ऑप्शन दिख रहा था हाइड का, तो हमने हाइड किया तो सब हाइड हो गया। ओके, नाउ इट्स, आई थिंक इट्स लुक्स गुड। So like if you see from our ancestor to now, from ancient to modern, so our microbiome is also changed from ancient microbiota to modern microbiota. Everyone knows that in ancient we use different type of diet and healthcare system. We was not dependent on the injections or medicine, processed medicine, chemical based medicines. We usually try to take the herbal product as a medicine and natural foods, and they was not processed. But now recent life we can see that we all using the pizza, burger, processed meat, and most of the medicine is also chemically synthesized. So you can see how much big shift is happens. Lifestyle, if we talk, so we was living in the natural houses made by wood or made by uh, clay or something. Now we are living in the concrete houses. So this also make the environment totally different, because now we are having the AC. Now it's a requirement of the filtered AC, which we call, it can filter the microbes as well. But before we don't have, and on the basis of this lifestyle, diet, healthcare, and living houses, our gut microbiota is also shifting from ancient to now into the modern era. So there are several microbes like bacteroids, firmicutes, ratio, Prevotella, Treponema. Like they was quite high, abundant in our gut or in our skin in ancient time, and now they these species are getting reduced. So new bacteria taking the place of these bacteria. So this modernization, how it's impacting to our health. So like, see, urban lifestyle, we have pollutions, everything made by the chemicals, processed food or freeze food. So this kind of things making the slow gut biosis. And this slow gut biosis make several complications like antibiotic resistance in our body, in our gut. So like everyone is talk about antibiotic resistance. That's why sometimes one antibiotic work once our microbes get the resistance, those antibiotic is no more used in that particular patient. So we need to shift. So that kind of is also challenge for the doctors to how many shift he can make. So in many countries, this kind of like antibiotic giving to the patients is also very restricted. It is not kind of primary care. So uh, without prescription, it cannot given. But like here, we can buy from the stores anywhere these antibiotics. So we have more resistance as compared to the worldwide other countries. And this dysbiosis also lead for the tumor formation, type 2 diabetes, obesity, and kids, infants is also like not away. They are also affected by this. So, like you can see, transmission of antimicrobials and sugars to the infants. So, in the ancient time, we was taking unprocessed sugars, crude sugars. So it takes time to digest, and along with sugars, several supporting components also comes. But now we are taking everything in the pure form. So this pure form is restricting several other genes to express in our body. So we are going like addiction. We are kind of addicted with several things. So how they impact, how this is happening. So okay, this cartoon I think will make you little bit understand. Uh, you can see this commensal bacteria as well as pathogenic bacteria living in the mucosal surface together, and they manage very well. But if any foreign particle like antibiotic comes, it doesn't know. It just know the bacteria. It doesn't know which one is commensal, which one is pathogenic. So it affects both type of bacteria. So this kind of effect lead to the dysbiosis in our gut. Okay, so maybe this slide will explain you very well what happens. So in balanced microbiota, so pro-inflammatory bacteria present, Treg inducing bacteria is present. Because both immune responses is very important for our health, and they are making the balance. There is a no disease development. So in dysbiosis, there is a two type of scenario happens. One is Treg inducing bacteria get reduced. In other hand, there is no affected, but pro-inflammatory bacteria get increased. So in both cases, this biosis is starts. So what happened? Treg number get reduced, T effector cell is increased. So balance is now disbalance. In this case, you can see Treg number is like before, but T factor number is increased. So in these cases, like there is an inflammation get started. So you people can get autoimmunity, allergy, or cancer development because of this biases. So maturing the immune system is also important. Making the balance is also important. Once balance is like dysregulated, then disease development starts. Okay. So how these microbes interact with our immune system? So like you can see that in this figure, our body has several lymphoid tissues, lymphoid organs, like mild, this is a mucosa related, NALT, this is a nasal, and GALT, this is a like gastrointestinal tract, and okay, BALT is respiratory, WALT is vulvovaginal, and SALT is kind of skin. So our full body has several lymphoid tissues and organs. So like here you can see the explanation of all this kind of associated lymphoid tissues. So how they are situated in our body. So I cannot talk all. I'm going through the gastrointestinal gal. So what is the function of these gal in our body? So they give the general immunity, prevent microbial invasion, underlying connective tissues, mediate immune response to organisms that do invade through the epithelium, prevent impair barrier by inflammatory responses. So this is an introductory slide for components of the gut because we are going to see in few slides how gut is connected with the immune system and micro antigen delivery. So here clearly you can see in this cartoon, this is the epithelial cell line, and just beneath there is a pairs patches. So next slide will increase some little bit more components. So as you can see, most of the antigen and these dendritic cell interaction is happens into the pairs patches. So these innate type of cells is circulated in mucosal layer, and this epithelial cell is also having some specialized cells. So there is a mucin secretion by the goblet cells. These are the goblet cells, and this is the innate intraepithelial cells. There is a epithelial stem cells, because in intestine always these epithelial cells is affected after digestion. This needs to be the renewed in some portion if damaged. So always these epithelial stem cells are active, and this blue one, intraepithelial cells. Uh, so and M cell playing role into the antigen delivery, and you can see some of the IgA secreted also in the lumen. These are the microbes, and you can see together antimicrobial peptides. So now we can go into the more detailed component of this gut. So like how so here included this is the pairs patch. Here we included mesenteric lymph node as well. So you can see intestinal lumen, mucosal epithelium, lamina propria, and mesentery. So most of the immune cells are present in this lamina propria. What happened in lamina propria? These pairs patch make the delivery of the uh, antigen through the M cells, and here dendritic cell capture the antigen. Either some antigen come inside, or they can make the dendritis to until the lumen. So if any antigen came near to the mucus, they can sample. And what happen after that? These dendritic cells can travel through the lymphatic drainage to the mesenteric lymph node. Like this, not only dendritic cell, other immune cells also can travel. So this is a kind of connection happens from the lumen, from the gut to the mesenteric lymph node, and then mesenteric lymph node is connected to the mainstream of the ducts, bloods. So these cells can circulate in whole body. And what happened here? These dendritic cells sample the antigen from the gut, present into the mesenteric lymph node, mature this T cell and B cell. These T and B cell then differentiate. Those procedure will I will explain in next few slides. And those mature T cells and B cells can circulate and affect, like can be detected in other portion of the body as well sometimes. So but lamina propria also provide the niche to sample for the B cell, because there is a three type of antigen presented cells: B cell, macrophages, and DCs. So, but proper introduction and plasma cell generation usually happen either in pairs patch or in mesenteric lymph nodes. And you can see this plasma cell can secrete IgA, and this IgA is divalent. They can secrete it out to the lumen, and they act as a neutralization for the microbes and microbial components. So this kind of mechanism usually in gall component and in gut happening to stop the invasion of any microbes inside the lumen or inside the body. So, see, in this cartoon clearly always happens. T cell cannot sample any antigen. So there is a specialized cell like dendritic cells, they can sample. That's why if you see any dendritic cells, always in cartoon also it's shown with the dendrites. So these dendritic cells sample from the lumen any antigen either from viruses or from the bacteria or from the fungi, and after process with the help of MHC1 or MHC class 2 presented to the T cell. So that's why always T cell asked to supporting cells APCs found anything, because without presentation these T cell cannot go further function. Okay, so like this is showing the little bit more on the cytokines secretion, how it happening. So as you can see, I always already explained DC is sampling the antigens, and recognition is happening through the TLRs as well, because DC also express the TLRs, epithelial also express the TLR. So after encounter any antigen, so they need to secrete some cytokines. So in case of tolerance signal, usually most of the APCs secrete TGF beta, IL-10 and retinoic acid. My major work was focused on the T-Rex cell, and for T-Rex cell generation, these three components either together or separate can affect for the differentiation of the T-Rex cells. And this kind of cells, if mature with some antigens and secreting this kind of chemokines, they are limited for the expression of pro-inflammatory cytokines like IL-4 or IL-17. Usually they IFN gamma or TNF, like they don't secrete. So because so like this is a pair patch area. So this dendritic cell recruit to the pair patch intact with the T cell. In the presence of TGF beta and IL-10, you can clearly see that there is a T-Rex cell generation. And these T-Rex cells can differentiate, proliferate, and act on the uh, on for the homeostasis and make the control on the pro-inflammatory responses. So once these Rex cell is differentiated, they secrete the huge amount of TGF beta and IL-10. Okay. So how this differentiation is started? Because there is a plenty of
Is it
antigen
a
out.
differentiation of T24, T4 bacteriophage?
T4 bacteriophage,
Is it a
interleukin
interconnected?
4.
Write your message Trilogy in chat box.
Okay, I will explain. So, like, see, there is a plenty of antigens in lumen, and always there is a delivery is going on from lumen to the mucosal layer. So, how this dendritic cell differentially recognize those antigen and give the downstream signaling. So, this figure is showing that on the basis of antigen, the differentiation and maturation of these dendritic cells can further differentiate these naive CD4 T cells into the different type of cells. So, like I explained, few antigen can act on the DC and secrete some anti-inflammatory signals like TGF beta and IL-10. They can make this naive CD4 T cell into T-Rex cells. But there are some other signals. So naive CD4 positive T cells usually comes from the thymus. They are kind of neither Th1, 2 or 17 or T-Rex cells. So on the basis of signal, they differentiate. Once they differentiate, it is very difficult to go either naive or interconnected. So now new research is coming that Th1 can convert into Th17 or Th2 and Th1. Uh, that phenomenon is called the plasticity. So, but there are several reports already shown that Treg and Ts17 interconversion. So, after differentiation, it happens, but in very low amount. So, as you can see, after differentiation of Th1, 2, and 17, they have different type of functions, different type of cytokine secretions. Like Th1 usually secrete IFN gamma. It function as a cellular immunity clearance of intracellular pathogens. Th2 usually most of the time it's differentiate into the small intestine, and those antigen usually from the food antigen, allergic antigens. So they give the humoral immunity clearance of certain extracellular pathogens. So this kind of differentiation is also important into the gut. Yes, 17 differentiation. So you can see here these cytokines are responsible for the differentiation. So they're secreted usually by the dendritic cells, and after differentiation, they are equipped for secretion of this kind of cytokines, IL-17, IL-22, and they involve into the tissue inflammation, autoimmunity, clearance of certain extracellular pathogens. T-Rex is known for anti-inflammatory responses, so it makes the tolerance and immune suppression. So pro-inflammatory responses is also important into the gut, so that we can clear the pathogens. But who is going to control those pro-inflammatory responses? So these T-Rex cells usually control those pro-inflammatory responses to suppress the immune responses, control the immune responses. Otherwise, there is a scenario of the start the damage of the our own cells because of this huge amount of pro-inflammatory cytokines. Okay, so now we will go more specifically. Until now, we talk about antigen and T cell differentiation through the DC. Now, what type of DC is regulating this kind of function? So, like one place I talk about retinoic acid or vitamin A. So, how this vitamin A, we always talk for our body, vitamin A is very important. So, vitamin A is a dietary component which convert into the retinoic acid through the DC, and these specialized. There is a not all type of DC can process this retinoic acid. There are few type of only DCs, which is called the CD103 positive dendritic cells. I will talk about this, how their differentiation, how they distinguish from other subtypes. So, like here, you can see different type of even though only single type of CD103 positive dendritic cell have several functions through the RA. If RA along with IL-5 and IL-6 comes, it can act on the B cell, and its differentiation only RA can make the T cell, and sometimes like natural FOXP3 positive T-Rex cells. So these T cells also convert into the T-Rex cell later on. Rh TGF beta induced FOXP3 T-Rex cells. So there is a two type of T-Rex circulate in our body. One is natural T-Rex cells, another is iT-Rex cell. Natural T-Rex cells usually come from the thymus. They are more stable, but induced T-Rex cells usually differentiate in the periphery, in our lumen, in our gut, or from any other mucosal surface, and they are less stable. They have quite high plasticity. They can convert into the other subtypes as well. Okay, so I think we are coming now to the data. So, like you can see, our intestine has several components: stomach, in small intestine, duodenum, jejunum, ileum, and then large intestine, which we call the colon. So, there is a maximum number of microbiota is usually present into the colon, and small intestine also present. So, as you can see that all the component of the intestine has different type of environment. Like stomach has most of the acidic environment and oxygen contamination, but small intestine oxygen contamination is getting reduced. pH is also controlled, and neutral pH usually present into the colon. So on the basis of pH, and digestion process is also food, in which form it enter into the stomach and in which form it's going out from the rectum. So food availability also vary from stomach to the anus. So on the basis of oxygen, on the basis of food availability, and these kind of things uh, control for the colonization of these microbes into the gut. So different type of microbes present in different compartment of this intestine. So like here I'm showing, so on the basis of that, regulation. Immune cell is also varies in small intestine, in SILP, is and in the MLMs. So as you can see, now here I try to distinguish all type of dendritic cell and macrophages. So they call the conventional disease CD103 positive and CD11B positive. So you can see there is a single positive CD11B positive, double positive CD103 positive and CD11B positive, and this is a single positive CD103 positive. So here we use one model, jump-free model, mice model. So we colonize single bacteria because in the pool of the bacteria you cannot understand what response is going on. So if you so this is a kind of commensal bacteria which can induce the Treg cell. This is a LPA, a control bacteria which cannot induce the Treg response but can give the pro-inflammatory response. So you can see that this CD103 positive, especially the DC, is varies from germ-free to BB, colonized, and to some pathogenic bacteria. So this is a macrophages, CH3CR1, CD11B, double positive cells. So you can also see little bit shift, but usually in this case we calculate the cell numbers. And this is a PDCs, BST2 positive, CD11C positive. So I'm not giving total detail and graph of the data, but as you can see that in MLM, the BB colonization increase the CD103 positive, CD11B positive bacteria, which is playing role into the tolerance. But LPA cannot give any significant responses. So same thing, like I'm not showing the bar graph, but you can see the data in SILP. Double positive cells get changes from germ-free to BB and BB to LPA. Not only this one, even macrophages and PDCs also. Usually PDCs cell number is very less, but they also have the very important functions. Okay, so if we more narrow down only CD103, because I'm going to talk about tolerogenic responses like Treg differentiation. So in that case, CD103 positive disease is playing very important role. So as you can see, this is the percentage representative figures. This is the macrophages. So how this disease and macrophages can distinguish. So few people say CX3 CR1 with some previous markers, they can express on the dendritic cell as well. So how they differentiate different type of antigens and give the downstream signaling. So if one antigen comes and only CD11B positive, CX3 CR1 positive DC can, if it recognize this kind of antigen, and if they secrete IL-6 and TGF beta together, it can lead to the differentiation of naive T cell into the TS17 type of cells, which has the function IFN gamma production, IL-17 production. If another antigen interact with the DC which has CD103 positive and CH3CR1 negative disease, and they secrete TGF beta as well as RA, they can differentiate into the Treg cell. So here you can see that our immune system is very clever and it has several subtype of dendritic cells. So they can recognize and sense the antigen and then they can act and they can differentiate. So main motto of showing this slide is that how pro-inflammatory type of T cell and how tolerogenic Treg cells differentiated in our gut, and they can make the balance and stop the any type of inflammation or infection together. Okay, so my one colleague, she was working on the how this retinoic acid is playing role. So here we analyze something like how this vitamin A is processed in our gut. So as you can see, vitamin A come as a retinol, while this kind of ADH and RDH and retinaldehyde, RALDH, these are the enzymes make this vitamin A in final conversion into the retinoic acid. So then retinoic is secreted out as well. So especially in the MLN disease, so what we did, we give only just antigen, which is like have the either germ free or antigen free diet. So only not bacteria. This is component like I already discussed. This is a component of the food. So if you give the food which does not have any antigen, so in that case their compartmentalization as well as DC cell number and percentage get affected. So in germ free, it has normal CHO. So you can see the percentage of DC from germ free to antigen free is varies. So particular this CD11B, CD103 positive DCs get increased. What happening into this? So this is a kind of one assay, fax assay we do in this kind of DC, and we can recognize that how much L flora is coming. So if there is a more L flora, it means that vitamin A to RA production is high. So in germ free, it is showing very high. Antigen free, which does not have any antigen, it's significantly low, suggesting that by this data, CD103 positive DC can process efficiently this vitamin A to the RA. And mostly other DCs also have, but this particular subtype of DC is playing this particular role. But on another hand, we tested CD103 positive, CD11B negative species that does not show any significant result. Okay, so does this kind of DC can induce the Treg cell? So we set up one in vitro experiment. So I always talk about dendritic cells. So now we can say that CD11C positive DC. This is a simple marker for the disease. So we isolated the DC from the mice, give any type of antigens. So like if we give the some tolerogenic antigen, in that case they produce RA and TGF beta. And if we co-culture this disease with the naive CD4 positive T cells, so they can differentiate into the iTreg cells. So how we will know in which division they are differentiating into the Treg cells? So for that we stain with the CTV stain. So this staining usually we do at the time of culture, before the culture, and we can track those cells. So this is the data you can see. In mock, there is a very limited Treg cell differentiation. Once we give the BP type of antigen, this is a commensal bacterial antigen, you can see FOXP3 positive Treg cells differentiated in every division. So this last usually we say that from here to division starts, and you can see until 4 to 5 peaks differentiation is going on, and this is a non-Treg cell. So definitely if you put cells into for the proliferation, they will divide. So Treg cells is also dividing, non-Treg cells also divide. If you give the some pathogenic antigen from the bacteria, so you cannot see any Treg cell differentiation. So this is a clearly showing that these diseases are playing role for the differentiation of the Treg cells. This we also call the de novo type of Treg generation, which is not coming from the thymus. They differentiated into the periphery. And we tested also in this positive positive T cells, they are also producing the IL-10, especially in tolerogenic bacterial infection. Suggests that there is a clear cut differences into the microbes, which can few can differentiate into the Treg cells, few cannot. Few has the pro-inflammatory responses. Okay, so to show this kind of therapy model into the mice, what we did, we sorted both type of Treg cells as well as T naive cells. So there is a one immune model system to develop the colitis. So we use the RAG1 mice, which called the lymphopenic mice, which does not have the B cell and T cell. So in this kind of mouse, if you inject naive cell, they can start the inflammation into the gut, especially in the colon, and they develop colitis-like symptoms. And in vitro, we differentiated Treg cells. These iTreg cells we injected into the mice in this along with the control T-Rex cells as well, which is isolated from the mice. And here we ask the question: does our T-Rex cells are functional or not? So that we can rule out that okay, this kind of T-Rex cell can suppress the inflammation response into the gut. So this is the experimental scenario. Okay. So this here you can see in colitis model usually people in the first line they measure the weights. So as you can see, T and alone reduce the body weight, and T-Rex cells you can see because we did not differentiate it natural T-Rex and T-Rex from the mouse. So as I explained already, natural T-Rex cells are more of the T-Rex cells, and these T-Rex cells have the phenotype of high IL-10 secretion, low interferon gamma secretion, and stop the antigen inflammation into the gut. So in I started from the mucosal and immune system. So there is a several other thing is also going on. So like this is another function in the gut mucosa in the Peyer's patch is happening. So any antigen which coming through the M cells to the Peyer's patch, they only not mature the naive CD4 T cell. They activate these cells, T helper cells, and it act on the B cell as well. So as you can see, B cell interaction, because B cell alone cannot activate. So these T cell activate to the B cell, and B cell can directly sample these antigen as well because it also a antigen presenting cells. In in this scenario, if DC comes and it also gives the another signal in the form of TJ beta and NO, so these DC can differentiate these B cell can differentiate into the plasma blast, and these plasma blast is specialized for the secretion of IgA. So you can see another loop is coming for the IgA secretion by these antigens, and these IgA is secreted out into the lumen, and then they control this kind of antigen delivery. They neutralize these antigens as well as the bacteria. So if we talk in more in detail, so our gut is so big. So now see, I narrow down even in the colon. This is a proximal colon. This is a distal colon. In proximal colon, antibody secretion is varies towards the distal colon. Like you can see in proximal colon, IgA bound microbes and IgG bound microbes. So on the basis of microbial diversity, all this is happening because microbes is vary from proximal to distal, and that make the changes into the IgA production as well, divalent, and which is secreted out into the liver. So as you can see, the ratio of ROR gamma T and Tbet positive cells is changed. Like in proximal comal TH17 and TH1 ratio is high, and clonal expansion is quite low. But in distal colon, microbial diversity get changed, and Tbet and ROR gamma cell ratio you can see it varies, and TH17 and TH1 ratio get down, clonal expansion get up. So you can see in proximal colon TH17 cells number is high, but in distal colon TH17 cells get reduced and TH1 cells take over. So all this is happening because of microbial antigen and its interaction with the immune cells, because they start the scenario to mature our immune system. Okay, so I think this slide will be going to be more clear how homeostasis is happening in the response of this IgA production. So you can see they also kind of another defense system, like I always say, IgA. So IgA production in homeostasis is quite regulated, and always it gets neutralized the microbes in the lumen, and there is a systemic dissemination and production of IgA and commensal tolerance Treg generation is happening. But in inflammatory bowel disease, which we can say that in mouse model we say colitis, and we can develop like by the DSS or by night B cell. Just now I talked about my immune system. So especially the Treg cell generation is affected in inflammatory scenario. So IgA and plasma T cells is quite
और कितने
dysregulated.
स्लाइड बचे हैं?
टाइम हो रहा है क्या? I think एक दो slide होंगे.
ठीक है प्लीज.
I will finish soon. Yeah,
यस
yeah.
यस.
So like you can see that this regulation of B cell maturation. In previous slide I showed BC sample the antigen, mature and differentiate B helper cells, and also interact with the B cell. And these B cell now play the responsibility for the IgA secretion. So if that scenario is dysregulated, then inflammation starts as well. So as you can see, IgA production is reduced in the inflammatory conditions, but IgG production get increased. Okay, so how they make a circle? This is a lumen, epithelial cells, lamina propria. So how? So still there is a question. Microbe is controlling the IgA production, or IgA is controlling the microbial communities. So microbes can modulate. There is a two scenario can modulate IgA synthesis and
कुछ
secretion.
वहाँ हवा आ रहा है क्या, या वाइड आपका हिल रहा है? वो जो
हमको
वाइड
लगता फैन लगा हुआ है. रुकिए हम फैन बंद करते हैं या फिर
कुछ अभी थोड़ा देर में वो ज्यादा हवा वो लग रहा है ना, जिससे वायर वगैरह हिलता है. वायर तो आप लगाए नहीं हुए ना, ईयरफोन वगैरह नहीं है
नहीं
ना?
नहीं नहीं ईयरफोन नहीं स्पीकर है हमारे पास.
ओ, बीच बीच में वो आ जाता है जैसे खरखराने का.
अच्छा.
ठीक है, कंटिन्यू कर दीजिए, कोई बात
हाँ हाँ कंटिन्यू करते
नहीं.
हैं.
जी जी
बस अब खत्म ही होने वाला है.
जी जी जी.
So I did not talk about mucus layer. Mucus layer is another kind of defense system which always secreted out by the goblet cells. So mucus layer restrict the microbial penetration into the mucosa lamina propria. Another is like I already says like there is a Treg cell function and this antimicrobial peptides, and now this is the IgA which is regulated by the microbes. Microbes itself is regulating the IgA secretion. So after sampling, these DCs make the mature B cell like through the T cell as well, like in previous slide I discussed, and this plasma cell secrete the IgA production, and this IgA production going into the lumen and control the microbial community into the mucus layer, and this remaining microbial community again give the signal, and this scenario is always going on. So by this way homeostasis and microbial community concentration is controlled into the gut. So they are controlling each other. So you can see IgA can influence gut microbial diversity and composition. So microbe is controlling the IgA production, and IgA is controlling the microbial diversity and its composition. So our immune system is self-regulating the microbes, and microbes also regulating our immune system in the way which way it go and differentiate. So like this, uh, yeah, this is my final slide. So as you can see, microbes is coming in our body. They interact with our immune system, give the signal several immune cells, and like they are working vice versa. So still there is a debate, and like always, already people know that microbes are very important part of our body, and now we cannot live without the microbes. So, uh, like
थैंक यू थैंक यू थैंक यू.
and
बहुत